Right. In physical method, in atmosphere, nitrogen react with oxygen. As a result, formation nitric oxide. Right? Again, this nitric oxide react with oxygen, nitrogen dioxide. Convert into nitrogen dioxide. Convert into nitrogen dioxide. What happened after lightning and tightening? When it is available rainwater, now this react with rainwater. Nitrogen dioxide with water convert into nitric oxide. Nitric oxide or nitric acid. Now it is converted into single bond. Now converted into single bond, and they convert finally as a nitrate. Finally convert as a nitrate. Already told you at very beginning, this triple bond nitrogen molecule cannot received by root. When it is converted into nitrate or nitrite,
then only plants can live.
This form, this is nitrite form. Nitrate form or nitrite form, or another form is ammonium form.
NH3.
Yes, sir.
And when it is converted into ammonium form also received. So mainly this nitrate or nitrite formation by physical method. In physical method, what happened during the lightning and thundering, atmospheric nitrogen react with oxygen, nitrogen oxide, and nitrogen oxide again react with oxygen, nitrogen dioxide. When it is react with water, HNO3 form. This HNO3 to split into nitrate or nitrite. Now in this formation received by this root. So I will explain later ammonia formation, how the ammonia formation by biological method. So anyway, through biological method also, with help of some of the microbes, these atmospheric nitrogen finally convert into ammonia. So anyway, in this three, any form received by plant roots.
Roots, sir.
Right, later they are accumulated in plants in various organs. It is act as a constitution part of mainly where nitrogen present in plant body. This constituent part of
Proteins.
right, proteins, only proteins.
Amino acids also, sir. DNA.
Both are same.
All,
the subunits of the proteins.
Yes.
Right, no need to repeat it.
Yes.
So protein are okay, protein, right, which made up of amino acids. So proteins and amino acids and another constituent what?
Membranes.
Membranes, cell membrane, phospholipids.
Lipids.
Proteins, okay. Phospholipids, okay. Membrane, right? Membrane, right? Another one. Membrane.
Cytosol also, no, sir?
Cytosol, no. Of course, in cytosol in the form of amino acids, okay, no problem. Already amino acid completed, right?
Yes, sir.
Nitrogen also
common,
present in nitrogen base also present in nucleic acids, right, sir?
Right, nitric, that means DNA, RNA, DNA
most
and
of the
RNA
plants
also.
have RNA as genetic material.
Right, it also completed. And another mainly the nitrogen present in?
Nitrogen mainly present.
Okay, right, protein, right, membrane, right, DNA, RNA,
NADPH.
Proteins, enzymes.
Okay, NADPH. What about chloroplast, chlorophyll,
Yes, sir. Chlorophyll.
Yes,
chlorophyll
sir.
pigment,
Yes, sir.
Yes, sir.
chlorophyll pigment.
Yes.
So mainly this nitrogen mainly constituent part of these all chemicals, right? Okay, is it clear? So anyway, after accumulated in plant body, from the plant body, it is transported to herbivores, right? Okay,
Yes, sir.
Yes, sir.
Yes, sir.
from herbivores to,
Carnivores.
okay,
Carnivores.
right, in biological cycle,
Carnivores
carnivores.
to top carnivores.
Primary carnivores to secondary carnivores.
Okay, next, secondary, next,
Tertiary carnivores.
Top carnivores, sir.
Saprophytes, sir.
law finally,
Saprophyte. Decomposers.
Yes, sir.
decomposers. From decomposers to
Atmosphere.
atmosphere.
Again in
Where
the
the atmosphere,
atmosphere, sir.
right,
Again, the
decompose,
cycle repeats.
Nitrogen
decomposer
cycle
to
repeats.
again atmospheric nitrogen, right? So this is like a cycle, right? So in atmosphere,
Yes,
first
sir.
one method is completed, physical method completed, right?
Yes,
And
sir.
Yes.
another one, Haber-Bosch method, industrial method, that is not an important one, but remember the Haber-Bosch method also. Another one in Haber-Bosch method, nitrogen with ammonium at the temperature around 400 to 600 temperature and pressure, I think 120 degree. I'm not idea surely.
Sir,
I think
121
120.
LB pressure, sir. 121 LB.
Right, 120 or 121 degrees centigrade pressure.
LB pressure, sir.
Right. So in this situation, ammonia formed in industrial level, right? So, but here mainly is the lightning only. Just right. But in lightning, biological nitrogen fixation, when compared with lightning, biological nitrogen fixation, the more. In lightning only 10 Tg, tera G for teragrams. And biological fixation, there is the 90 to 140 teragrams per year. So mostly the nitrogen fixation done by biological method. Clear? Let's see biological method. Already some brief introduction, the last class about biological method. Should I explain this? All nitrogen fixing organisms, bacteria, cyanobacteria, symbiotic, legume, non-legume. Hello?
Rhizobium bacteria.
Yeah, they're all important. To page number 240. Bacteria, let's
It's
see,
okay, sir.
aerobic bacterium. Aerobic bacterium, example, azotobacter. One example enough. One example
Okay.
enough for one type.
From each category, one.
Yes.
Okay.
Aerobic bacteria, one. Azotobacter, enough.
Azotobacter.
Micro aerobic bacteria, one.
Azospirillum.
Azospirillum, enough. Facultative bacterium, Klebsiella.
Pneumonia. Yeah.
Non-photosynthetic, Clostridium. Photosynthetic nitrogen fixation bacteria, Rhodospirillum. And cyanobacteria, in cyanobacteria, unicellular nitrogen fixation, Cleocopsa. Filamentous heterocystus, nitrogen fixation, cyanobacteria, Anabina. And filamentous non-heterocystus, nitrogen fixing, cyanobacteria, Plectonema.
Oscillatoria also we can write some.
Oscillatoria also given, but I think it is not involved in nitrogen fixation, as per my knowledge.
Okay, sir. So Plectonema is the one we could
Best one.
go with. Okay,
right.
sir.
And symbiotic, well known to all, the legume symbiosis, Rhizobium bacterium.
Yes, sir.
Non-legume symbiosis, Frankia, enough. Frankia in Casuarina plants. Legume, which plants? Leguminous family. Here given
Beans.
glycine marks.
Yeah.
Legumes, no need to dissolve. Just write down legume plants. Enough.
All right, sir.
Right. And symbiotic nitrogen fixation. Symbiotic nitrogen fixation, no need to explain this all. Asymbiotic nitrogen fixation, azotobacter, cyanobacter, already given in table clearly, but no need to elaborate it. But symbiotic nitrogen fixation here, legume plants. Stop. Right. Symbiotic nitrogen fixation. Nodule legumes and another one cyanobacterial symbiosis. First one legume rhizobium bacterium. Well known to all rhizobium bacterium. They are soil-borne bacterium. These are all
gram-negative bacteriums.
right.
Soil bacterium.
Soil bacterium, gram-negative. Okay. How they are attracted? Already explained the last class.
They release some kind of
Yes, sir.
liquid
Like a flavonoids and the lectins.
Flavonoids, lectins. First, before
Yes.
accumulated this bacteria from the host plant, here some flavonoids,
Flavonoids compounds and then lectin.
Flavonoids, FN flavonoids and lectin chemicals released from the host plant. From this chemical, the bacteria attracted and accumulated near the root tip. The next what happened? This root
hair curl
shows curved like a hook-like structure.
Curls up. Yes.
This is called shepherd crook. This nature called as shepherd crook. After that, what happened? The same way as a group, this bacteria, this rhizobium bacteria. Please be silent. Hello, please. Thank you. Right. This rhizobium bacteria enter through the rootlet. This is called as rootlet. Small rootlet. Enter through the rootlet as a group. Invasion into the cortex cell through the epidermal layer and settled in cortex. Where after entering the cortex cells, automatically this bacterium active the auxins and cytokinins. Auxins and A for auxin and C for cytokinins in the cortical cells. Due to the activity of this auxins and cytokinin, these cells, cortical cells, rapidly redivided and form as a group of cells. This is called root nodule. Like this, root nodules are formed. Okay, then how the formation nitrogen? How the fixation of nitrogen, atmospheric nitrogen? After settled down inside the cortex cell, what happened here? Bacteriaids formed. Actual rhizobium bacteria first. Rhizobium bacteria or for rhizobium bacteria, when enter into the cortical cell, it is converted into bacterioid. Bacterioid means simple. This rhizobium bacterium instead of cell wall, bacterial cell wall, the extra layer covered with help of this infection thread. This is first actually the infection thread emerged out. This infection thread, like for example, this is infection thread. Through infection thread, this all bacteria, let me change color, all bacteria in blue color, bacteria enter into the cortical cell. Later, this infection thread covered around the bacteria as a layer, covered as a layer. So now each bacteria blue color bacteria, B bacteria covered with infection thread. Now this is called as bacterioid. Now this is called as bacterioid. When the bacterioid form, now it is bacterioids. After bacterioids formed, now ready for atmospheric nitrogen fixation. Right? Is it clear?
Yes, sir. Yes,
Right.
sir.
So this root nodule formation under control of NOD genes, NOD, NOD genes. These NOD genes activated in bacterioids. NOD genes activated in bacterioids. After NOD genes activated, immediately leg hemoglobin 14 formed inside the bacterioid. Between this membrane and bacterioid, here, let me change green color, leg hemoglobin formed. Leg hemoglobin formed. Right after formation bacterioid, through the transcription, I will explain later, transcription translation process, the NOD gene proteins are formed or NOD genes activated. So under controlling of NOD gene, nitrogen fixation takes place in plant body, in legume plant body. So far clear? Hello?
So could you
Yes,
repeat
sir.
that once again, sir, please?
First bacteria formation clear?
Yes, sir. That is clear.
Yes, sir.
After formation
Bacterioid formation.
That
this
also.
bacterioid,
Yes, sir.
Yeah.
leave it leg hemoglobin. This bacterioid forms the activate the NOD genes.
No, not genes.
NOD genes.
Okay, sir.
Right?
Yes, sir.
Later leg
Yes, sir.
hemoglobin formed. Leg hemoglobin
Between
formed in
these layers.
bacterioid.
Okay.
Yes,
Between
sir.
the layers, sir?
Right.
Okay.
So this leg hemoglobin, it is a pink colored compound and it is more or less similar to mammalian hemoglobin.
Like
Right?
a blood
Almost.
hemoglobin.
Right. Like
Okay.
a blood hemoglobin, but variation in amino acid composition.
Okay, sir.
Like hemoglobin protein, like hemoglobin also protein, any protein made up of amino acid, but arrangement different.
Okay, sir.
For some cereals
Okay,
same.
sir.
That's why it is more or less similar. And it is located between envelope and bacterioids.
Okay,
Between
sir.
envelope and bacterioids. So here very interestingly, leg hemoglobin, heme portion,
That is iron, sir?
no, actually leg hemoglobin formation, heme part and globin part.
Heme is iron, no, sir?
Heme, some protein.
Iron protein, or it's only a different
Right,
type of
iron
protein? That's what I'm asking. Sir. Thank you, sir.
Right, it is synthesized by bacterioids. Heme by bacterioids. Globin from host. Globin formation. Globin protein part. Globin protein part from the host. Globin protein part from the host. Now both together form as a leg. It is formed in Leguminosae family. That's why, and it is resembled to the hemoglobin. That's why named as a leg hemoglobin. Right?
Okay, sir.
Right. Now what is the function of leg hemoglobin? Why it is formed? Actually, this nitrogenase enzyme, remember, nitrogenase enzyme synthesized from the NOD genes. NOD genes synthesize nitrogenase enzyme. Nitrogenase.
Sir, it takes sugar from the plant, sir.
Which one?
Leg hemoglobin function.
Repeat it.
It takes sugar from the plant roots.
Leg hemoglobin takes or bacteria takes sugar?
Bacteria takes, sir, with the help of this.
Right. Actually, these bacterioids do not use any nutrition. They are stable. Only rhizobium bacteria utilize the sugars. But after convert into bacteriaids, no utilization of nutrition from the host. Only before formation, bacteriaids utilize the host, what you said, that is sugar and whatever it may be utilized. But after formation bacteriaid, it is completely covered with layer, right?
So is it appropriate to call
Yes,
this
sir.
leg hemoglobin as pigment, sir? No.
Leg hemoglobin pigment.
No, I don't think so. Is it? Or we can call it as a compound. What would be the appropriate term, sir?
Leg hemoglobin, maybe it is.
Because for a
Yes,
mammalian hemoglobin, we call that as a pigment. No, sir.
The pink color, so it's also
Yeah.
pigment.
Yeah. So hemoglobin, leg
What is the
hemoglobin.
first sentence? Even filled compound. Leg hemoglobin is a pink
So we
color.
can call it
compound.
as a compound.
Is it just wait? Is it a pigment? Pigment leg hemoglobin present. Okay, it is pigment.
Yes, sir. Thank you, sir.
Pigment compound. Okay, right. So nitrogenase enzyme synthesis from the NOT genes, right? They mainly this nitrogenase enzyme after formation nitrogenase enzyme in plant body, rhizobium bacterium, then only fix our atmospheric nitrogen into ammonium. Atmospheric nitrogen into ammonium. Right. So what is the function of your leg hemoglobin? Why it is required? What is the requirement of leg hemoglobin? What is the importance of hemoglobin?
It carries oxygen.
Same like that, here the leg hemoglobin controls the concentration of dissolved oxygen. Because if it is this nitrogenase enzyme, oxygen sensitive. If oxygen present inside the cell, this nitrogenase enzyme not active. Not active means actually these NOT genes cannot synthesize the nitrogenase enzyme. These NOT genes active only absence of oxygen. These NOT genes only synthesize nitrogenase enzyme absence of oxygen.
So only in the absence of oxygen, the NOT genes can synthesize nitrogen. Is that so?
Right, nitrogenase.
Okay, sir.
When nitrogen is present, should not present oxygen molecule inside the cell. If any oxygen molecule present, it react with nitrogenase and deactivate immediately. So it is not possible to fixation atmospheric nitrogen as a ammonium. Clear?
Does it mean, sir, if the oxygen is there, some other compound is formed instead of nitrogenase?
Some other compound means?
Maybe nitrogen dioxide or something like that?
No.
Okay.
Actually, during respiration, what happened? Oxygen released, right?
Yes, sir.
This oxygen diffused, actually this takes place in leaf mitochondria.
Yes, sir.
Mainly mitochondria in all cells and diffused into each and every cells. Right?
Correct.
Like that in root cells also diffused.
Okay.
But when the formation, after formation of leg hemoglobin, immediately capture the oxygen.
Okay.
If leg hemoglobin absent, what happened?
There won't be any oxygen uptake.
No, for example, this is the cell, right? So this is the cell.
Sir.
Like any other cells here also, if oxygen present, this is one of the cortical cell of the rhizobium.
Yes, sir.
Oxygen present. Actually, if leg hemoglobin present, leg hemoglobin, LHB. If leg hemoglobin present, already told you the function of hemoglobin carry oxygen, right?
Correct,
that means
sir.
react with oxygen.
Yes,
so in the same way here also it is react with oxygen, that means capture the oxygen.
It's an oxygen carrier. Yes, sir.
Right, oxygen carrier, and it completely take away.
Okay, it carries away from the
Cell,
not.
existed cell.
Okay.
So
Carries
the
away.
rhizobium is itself is anaerobic bacterium, right, sir? It cannot survive in the presence of oxygen.
Rhizobium,
Itself is anaerobic bacterium.
sure.
That's why it cannot survive in the presence of oxygen.
Aerobic or anaerobic?
Anaerobic.
Rhizosphere, aerobic.
Rhizobium is a legume symbiosis. Sir.
Rhizobium,
Yeah.
here the anaerobic bacterium only Clostridium, I think. Let me see here. The page number, what is it? Where the table?
240,
240.
sir.
Anaerobic bacteria only aerobic, microaerobic, facultative.
Only aerobic one is given, sir.
Only aerobic. Anaerobic bacteria, microaerobic, photosynthetic. Actually not given here. Anaerobic bacteria, I think.
Sir, if one is given as aerobic means remaining all are anaerobic.
Anaerobic bacteria. That means anaerobic nitrogen fixation bacteria.
Anaerobic means it cannot survive
Sir,
in the presence of
actually,
oxygen, right, sir?
Clostridium has given somewhere. Clostridium given. Clostridium non-photosynthetic. Clostridium non-photosynthetic and anaerobic nitrogen fixation. Clostridium species for non-photosynthetic and anaerobic bacteria. Anaerobic nitrogen fixation. Right down there.
The rhizobia are gram-negative bacteria. They have aerobic life, sir.
Right, aerobic. That's why I'm in confusion.
yes, sir. Rhizobium is an aerobic, sir.
It is a anaerobic bacteria, but as of knowledge, it is a aerobic bacteria.
Yes, sir. Rhizobium lives as an aerobic microbe.
Right, aerobic bacteria. But in the presence of legumoglobin, what happened? Oxygen completely.
It devoids of oxygen. It doesn't
Right.
require oxygen.
Right.
So hence the leg hemoglobin is needed to carry the oxygen from the nodules.
Existed cells.
Okay, sir.
Right. Now then only these NOT genes prepare the nitrogenase enzyme with help of nitrogenase, atmospheric nitrogen convert into ammonia.
Okay,
is it clear so far?
Okay, so that is the function of leg hemoglobin.
Right.
Thank you, sir.
Now next, how it is convert mechanism. Let's see the mechanism of atmospheric nitrogen convert into ammonia. Can't we take the picture from the desktop?
You can, sir.
But where share screen, whiteboard, iPhone.
Directly you can share the screen, sir, on your desktop, sir.
Are you able to see?
Not
No,
sharing
sir.
as of yet, sir.
Now?
Yes, sir, can be seen.
Right. So nitrogenase enzyme. What are the components of nitrogenase enzyme? Already given in your book. Nitrogenase enzyme. Nitrogenase. Made up of mainly what are the subunits of nitrogenase enzyme? Right, in page number
244, I think.
246.
246. Yes, sir. Large protein contain amino.
Right, it is the
and
nitrogenase
iron.
consists two different protein. One is large protein part and small protein part. And here the
Yes,
large
sir.
protein contains molybdenum 2 moles, molybdenum. And ferrous. Molybdenum and ferrous. And sulfide also. Molybdenum, ferrous and sulfide. And smaller one, only protein pods. Only smaller one contain protein pod. Right? So
Smaller protein contains 4 iron and 4 acid.
And sulfide also.
And
right?
sulfide acids. Yes, sir.
Right.
Yes, sir. Acid level sulfide.
Protein, molybdenum and sulfide. So this is nitrogen enzyme, right? So first protein part, molybdenum. Right. So this together, ferrous, molybdenum and sulfide together called as
Holo enzyme.
completely. That means this completely together forms the nitrogenase enzyme. Right?
Yes, sir.
Yes, sir.
Right. Okay. First, where the electron donor? For reduction, what is reduction process? That means actually atmospheric nitrogen reduced into ammonia. That means in this process, it should be received by electron.
Yes, sir.
Right here, the electron donor is ferridoxin. Ferridoxin, electron donor. Have we heard this word ferridoxin? Hello?
Yes, sir.
Tell me.
Sir, it is the compound of ammonia.
No.
Compound of ammonia. Ferridoxin.
I heard of it, sir, but I'm not able to recollect it.
You did not attend on that day. Bambina.
Yes, sir.
You did not attend on that day.
Offline class, sir?
Yes, offline class.
Yes, sir. I didn't attend. I remember
Sir, I too,
vaguely,
sir.
long time back, I have heard of it. I'm not
Ferridoxin.
able to.
It is compound of iron.
Okay. Ferrous, sulfur, but where it is?
Ferridoxin.
In which reactions?
Sir,
Appeared this word.
photosystem 1.
Right, in photosystem. In photosystem, phosphorylation. In light reaction, what is the final electron recipient?
Oxygen.
Yes.
Yes, sir.
FD.
Ferridoxin gives the electron to
yes,
final
sir.
recipient such as NADPH.
Yes, sir. Yes,
sir.
sir.
Right? So
Yes.
like this here
Yes,
also, here from the ferridoxin, the electron donor. Right?
Yes, sir.
From electron, when electron loss from the ferridoxin, it undergoes to oxidized state. Oxide.
Yes,
Now
sir.
this electron received by nitrogenase ferrous As it received, it is undergoes to reduced state, right? And again lost this electron and again converted into oxidized state. And this losing electron, always 2 electron, remember. This electron from the ferredoxin, from the ferrous to received by molybdenum. Now molybdenum reduced. Molybdenum ferrous reduced. Now the reduced form of this molybdenum ferrous, loss of 2 electron, and this 2 electron gives to the
nitrate.
NH. I think it is nitrogen amide, something name. NH formed. And again, extra 2 electrons received the same way. Amide. NH2, single bond, NH2. And again 2 electron convert into ammonia. Like this ammonia formation in legume plants. Just wait, somewhere it is stored. Diagram. NH2, NH2. Just wait here. Are you able to see the screen?
Yes, sir.
Yes, sir.
This one clear?
Yes, sir.
Cycles. Nitrate detectors, ferredoxin.
No, sir.
Are you able
No,
to see
sir.
this mark?
It
Arrow
No,
is,
sir,
mark?
the PPT only we are there, sir.
Okay.
Working
Okay.
enzyme of
working of
nitrogenase. That's
enzyme
it.
nitrogenase.
Bah. Where is previous slide? Okay. Okay. Simple here. First ferredox, electron donor, ferredoxin, right? Are you able to see
Yes, sir.
FD.
screen?
Yes, sir.
Yes, sir.
Yes, sir.
Ferredoxin. Always the ferredoxin donate 2 electron. After loss this electron, it is converted into oxidation state, right? So this electron received by
Yes, sir.
Fe protein, ferrous of nitrogenase enzyme. Already told you the components of nitrogenase, ferrous, molybdenum, and sulfide, right?
Yes, sir.
Now this, sorry, right. After received this electron, it is undergoes to reduced state. From this ferrous, this electron, 2 electron lost and received by molybdenum ferrous protein. As a result, it is reduced. What about the Fe protein? After loss of 2 electron? After loss
It
of 2
becomes
electron?
oxidized.
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 Right? Now reduced MO,
Yes, sir.
molybdenum Fe protein, same 2 electron sent
And
to
becomes
the
oxidized.
HN double bond HN. Diamide, something, the name, I think it is called diamide. Is there given any name? Just wait. Actually not given in our book, but let me see.
Yes, sir.
Diamide, I think. Just wait. Anyone have intermediate second year book? Intermediate second year. Oh. Just wait.
Which book you want, sir?
Intermediate botany second
I'll
year
just
book.
check, sir. My daughter might be having. I'll just get it from her bag.
Okay, just keep in mind. We'll check later. Okay.
All right, sir.
But the chemical name I
Okay,
don't know,
sir.
but I think amide, diamide. I think it is amide. NH2 diamide, and final ammonium. For in each step, 2 electron.
Diazine diamide, sir.
Right. 2 electron sent.
Yes.
Right. So in our book, given the reaction, nitrogen plus 8 electron plus 8 protons, and total ATP, utilized ATP number 16. From each
16,
step,
sir.
Yes.
here
16.
2 electron
2.
missed.
So total 6 electrons. Okay.
1, 2, 3,
6 electrons are going.
4. Right. 1, 2, sorry. 1, 2, 3, 4. Total 8 electrons
8.
plus, but in some books given 6 electron, but remember 8 electrons
8
and 8
protons.
protons.
Okay, sir.
And utilized some same 4 steps, right? 4 into 4, here 4 ATP. For each and every step, 4 ATP utilized.
Hmm.
So here from each step, 2 electron, 2 electron, 2 electron.
Okay.
From each step, 2 proton, 2 proton, 2
Yes,
proton.
sir.
Here from each step, 4 ATP.
Yes, sir.
That's why 4 into 4.
4 ATPs.
4 into 4 total ATP.
16.
16 ATP. Total
16,
16.
sir.
16 ATP. Utilized. That means 8 electron, 8 proton, 16 ATP utilized for formation of
ATPs.
2 mole of ammonium. 2 mole of ammonium.
Yes, sir.
Right? Is it clear?
Yes, sir. Yes,
Right.
sir.
So this is mechanism of nitrogen fixation. Right? So anyway, finally ammonium formed. And this ammonium accumulated in plant body in the form of DNA, RNA, chlorophyll, and what it may be, plasma membrane. And another one, what you said earlier, constant portion of nitrogen.
Nucleic
Chloroplast.
acid.
Right, nucleic acid completed. Nucleic acid,
Chloroplast.
plasma membrane, chlorophyll.
Chloroplast.
Chlorophyll,
Right,
sir.
these are all.
Yes, sir.
Right?
Yes, sir.
Now, right, we are the reduction of nitrate to nitrite. Nitrate reduction. What is nitrate? NO3
NO3.
NO3, sir.
Yes,
minus.
sir.
Reduction means receive electron, right?
Yes, sir.
Yes, sir.
So after received electron, nitrate converted into
HNO3.
NO2, nitrite.
Nitrite.
Right,
NO2,
sir.
sir.
Nitrate converted into nitrite.
Nitrite, sir.
Yes, NO2.
Right. So this is nitrate reduction. So this reaction, nitrate reduction or nitrate reduction also, nitrate in it, when if it is
Reduction.
received another electron, convert into nitric oxide.
HNO3.
NO3, NO, only NO.
Okay.
Nitric oxide, right? So this is mainly takes place in this reduction of nitrite, mainly in cytosol of shoot, root, and any other vegetative parts, right? So nitrate reduction mainly, mostly this nitrate. That means here in physical manner, nitrate formed in the form of nitrate after received by plant body. In plant body, it is stored in the form of nitrate. But inside the plant body, after accumulated in plant body, inside the plant body, received another electron and converted into nitrate and separated into finally nitrogen. And this nitrogen, after separated, this nitrogen utilized for whatever it may be, DNA structures, RNA structure, plasma membrane structure, any other material, preparation in any other material, right? In the same way, this ammonium also, after formation into single bond like this, inside the plant cell or inside the plant cell in this single bond separated H plus and utilize this direct nitrogen and involved in constitute part of the enzymes or proteins, any other proteins or enzymes or DNA, RNA, like this. So here the no need to explain about this complete structure and the regulation of nitrogenous gene expression. Already regulation I have explained in single word. How it is, what is gene expression? Hello? What is gene expression? Gene expression, that means expression means here what?
In
Express the character.
enzyme synthesis, sir.
Okay, enzyme synthesis, that is the regulation.
Regulation.
Expression.
It
Here
express its
given
character.
regulation of NR gene expression.
Express its character, sir.
Yes,
Shows the
expression.
character.
Right, expression is the character. When the NR gene, nitrogenase NR gene or NOD NOD gene synthesize nitrogenase enzyme, when it is regulated, when it is switched off, when it is switched on. Already given clue. When nitrogenase enzyme in active state, when nitrogenase enzyme active state and convert atmospheric nitrogen into ammonium. Presence of oxygen or absent of oxygen. Hello?
Absence, sir.
Absence of oxygen. In absence of oxygen, not genes prepare, that means not genes switch on and prepare nitrogenase enzyme. When presence
Correct.
of oxygen,
They become inactive.
become inactive. I will explain clearly next class in protein synthesis. Okay?
Okay, sir.
Right.
Okay, sir.
Right. In next one, biosynthesis of protein and genetic code. Let's see biosynthesis of protein and genetic code. Right. right? Genetic code table. Before biosynthesis of protein, what is biosynthesis of protein?
Preparation or synthesis of protein, sir.
Are you able to see this table?
No,
No,
sir.
sir.
Just wait, just wait.
Yes, sir.
Right?
Okay.
Right?
Yes,
Yes,
sir.
sir.
Right, in protein synthesis, already you might have known about this. Central dogma. What is central dogma theory? Crick central dogma theory. We proposed in 1955 central dogma.
Sir, information from DNA to RNA, sir.
Right.
And also to the protein.
DNA in genetic information carry forward to RNA, RNA to protein.
Yes, sir.
RNA to protein. So this is called central dogma. These proteins are all called enzymes. When this enzyme prepared, these are react on appropriate substrate. Then only character expressed. That is called gene expression. Simple, this is the cell. Be careful, listen carefully. This is the cell, right? Inside the cell, flowering enzyme, F, this is flowering gene. Flowering gene present. But this gene character not expressed until when it is reached reproductive stage. Until then, it is in switched off state, right? Hello? Are you follow me?
Yes, sir.
Yes, sir.
Yes, inside the cell, from the beginning of organism, or from beginning of the plant, F gene, flowering gene present. But when reach with reproductive state, this flowering gene active, and this is for gene. This gene prepared RNA, right? From RNA to protein. All proteins are enzyme, right?
Yes, sir.
Now this protein react on substrate. Which substrate? Flowering substrate. Flowering substrate. Actually, this flowering substrate formed when it is reached reproductive stage. After formation substrate, immediately the flowering gene, DNA of gene activate and prepare RNA and protein, then only the one character expressed. Is it clear? Hello?
Yes, sir.
Can I give another example?
Yes, sir.
Yes, sir.
Best example, human beings.
Okay, sir.
And you give me example. That means any character. White hair formation.
Yes, sir.
Or diabetic.
Okay, sir.
Diabetic. Actually, your parents, someone, your father or mother, anyone already suffering from diabetic.
Genetic.
Okay, sir.
Some genetic problems, and that means already facing the problem with diabetic. The
Okay.
same gene from your parents, you carry the same gene. You carry the same gene from the beginning, from your childhood. But when you in childhood, the diabetic character expressed?
May or may not
No,
be,
sir.
sir.
That means before 30 years, before 40 years, this diabetic character, of course,
It
you
will be
maintain the
resistant.
healthy diet
It
and
will be
proper exercise.
suppressed probably.
So no,
Even in
sir.
this, in this situation also, after 40 years, you will get diabetic. What is the reason? What is the reason here?
Probably that is getting activated at
Yes,
that
after
time.
40 years, that gene activated and express the character. Simple. Hello?
Yes, sir.
Right. So
Yes,
these
sir.
all genes, right? So in DNA, carry forward to RNA, but is in RNA, they are in triplet form. Triplet. They are in triplet form. Already told you why they are triplet form, because in actually in complete DNA, only 2%, no, only 5% genes are active within 100. Within 100, only 5% active. 95% junk gene. That means this junk gene does not code for any triplet codons. Only 50% codes amino acids and codes protein. The 95% does not code. They are all called junk genes, right?
And what
What
is the
is
function, sir? Junk gene?
junk gene function?
Yes, sir.
Actually, junk gene function for that mean, do you know the nucleosome model chromatin structure? It
Yes,
is
sir.
involved in chromatin structure. That
Okay.
means they give the stability to chromosome.
Okay.
Only stability chromosome means, do you know how much length of the DNA? How much length of the DNA stand?
No, sir.
Gene completely, length
Gee,
of the gene
there's one term, sir.
in kilometers.
Okay.
What about your height?
It's all because of the genes.
I'm asking the length of the DNA, length of the gene length. Gene length of an organism in kilometers. But what
Okay.
about your height?
It's
Just
in
feet.
meters. Meters,
Just
feet.
feet.
Yes.
How it is compact? What about the cell? What about the nucleus?
Microns, sir.
Very microns. But when you expanded DNA, it is expanded up to some of the kilometers. But such a length of the DNA, how compacted within tiny cell, within minute cell,
Yeah.
because of folding, compactly folded.
Yes,
This
tightly.
compactly
Yeah.
folded with help of histone protein
Yeah.
and with help
Okay.
of some junk genes.
Okay.
Some
Okay, sir.
examples ఇచ్చాను చూడండి, ఒక అగ్గిపెట్టెలో
Okay.
చీరని పెట్టారని.
Yes, yes.
సేమ్ ఆ
So
ఫార్ములా అంటే
that
అంత
helps in making it compact,
very compact.
packing it up. Okay, sir.
Right.
And
Clear?
this 5% of it is helping in coding
Formation
the
this
protein.
like this triplet.
Okay.
Like this triplet. So what about how many nitrogen bases? How many nitrogen bases?
4, sir.
4, sir.
4. In RNA, instead of thymine, uracil,
Thymine, uracil will be
cytosine,
present, sir.
Yeah.
adenosine, guanine.
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 Right. But how many triplet codons are present in an organism? 64. 64 triplet codon. This triplet codon formed by only these 4 nitrogen bases.
Yes, sir.
From these only 4 nitrogen bases. From 4 nitrogen bases, how become 64? And the 64 nitrogen bases codes for how many amino acids? How many amino acids present in cell? How many types? 20 different types. 20 different types amino acids present.
20 essential amino acids, right, sir?
Right. So remember, only 4 nitrogen bases become 64 triplet codon. The 64 triplet codon codes for only 20 amino acids. These 20 amino acids together form a different form of proteins. Different form of protein. Let's see how these 4 nitrogen bases converted into 64 nitrogen bases. Sorry, 64 triplets. Here,
Yes, sir.
uracil, cytosine, adenine,
Adenine,
guanine.
guanine.
The same column, take horizontal line here also, uracil, cytosine, right? This is 1 block, 2 block, 3 block, 4. Total 4 block, right?
Yes, sir.
Right. So for each block, same again, you have to take 4 nitrogen bases. Okay?
Yes, sir.
Uracil, uracil, uracil. Are clear? 1,
Okay,
2.
the same
Yes,
from
sir.
here, all the 3 sides, you need to put it together.
Right.
Okay.
And again, here U common,
C
C,
U.
and U.
and again U.
Yes.
U, A, U. U, G, U.
U, G, U.
Right. U, G, U. This is common for everyone. U, G, U. U, G, U. And the same way U, U, and second C. So in second column, last one is always C, right?
Yes, sir.
And third one, last one always A, right? And fourth
Correct.
one always G, right? Like this,
Middle one also, sir.
right? As
The
it is
middle
same.
one, what is there on the top, the middle one will be there.
Yes, exactly. When you count all these 64.
Okay,
In
sir.
64, you must have learned about these nonsense codons. 3.
Stop codons, right, sir?
Yes, stop codons nonsense. UAA, UAG,
UGA.
UGA. These 3.
Yes, sir.
Okay.
And remaining 61 codes for amino acids.
Okay.
So here, how many amino acids here? 1, 2, and
20,
again
sir.
repeated. And again repeated. Okay?
Yes, sir.
3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17,
22.
18,
20,
19.
sir.
Only 20. Someone actually some of the
22.
amino acids
Repeat.
repeated.
Repeated.
Are repeated, sir.
Yes,
Yes.
sir.
So only
ARG,
20 amino
ARG
acids.
has been repeated.
Arginine.
Right. Arginine, right.
Arginine is repeated.
Arginine
Yes.
and leonine, this also repeated. Okay, so
Sir.
this is genetic code. Clear?
Yes, sir.
Obel hypothesis, right,
Yes,
sir?
sir.
Obel.
Yes.
Obel
Obel
hypothesis.
hypothesis means last codon. That is something.
Stop codons, sir.
No. Obel hypothesis, that is not. Obel hypothesis means in this triplet codon, last to one, that means ఏముంటదండి?
Opal, ochre, amber.
No. Just wait. Last one not involved in 1, 2, 3. This is not importance. The last one not important because this is uracil. This is uracil. What are the decided factor here? First 2, right? Hello?
Yes, sir.
Yes, sir.
Only first 2 decided factor to recognize proper amino acid.
Amino
So
acid.
last one no use. So that is called as Obel hypothesis, right? Okay. That is called Obel hypothesis. Okay?
Sir, is the methionine is the start codon? Can we say it? Can we
Yes,
call
methionine. Now I will start the transcription translation. This is
Okay,
only
sir.
genetic code.
Okay, sir.
Okay, sir.
Okay.
Yes,
In the
sir.
next class, protein synthesis, transcription and translation both.
Translation. Okay, sir.
Right. Any doubt? Any doubt, ask me. I will give 2 minutes. Transcription eukaryote, transcription in prokaryote, but you are only in your book given only transcription prokaryotic transcription and prokaryotic translation. But we will discuss both transcription in eukaryote and prokaryote. Translation in eukaryote and prokaryote. Just comparison. Okay?
Okay, sir.
Right? Nitrogen metabolism, nitrogen fixation, biological nitrogen fixation mechanism, all are clear.
Yes, sir.
Yes, sir.
Right? In this important one, actually, oxidative phosphorylation important. But in protein synthesis, nitrogen fixation cycle important. But in nitrogen fixation, not SA1. Nitrogen fixation, most probably short answer. Right.
Okay, sir.
Biosynthesis of protein, only genetic code, short answer. Genetic code is just actually not given this table in our book. You have to draw this table and write about this. Short answer.
Will it come, sir? Expected
Yes,
question?
this is also expected question.
Okay.
For short answer.
Okay, sir.
Okay, sir.
And central dogma theory, that means transcription, separate question, translation, separate. These both are.
Sir, do we need to write that entire table for genetic code, sir, if it comes for short question?
Short, even it is short question, you have to write all. That means you have to draw the table.
Yes, sir. Okay, sir.
This table we have to draw, sir.
This one enough.
Okay. So based on this
Sir,
table, we need to explain.
That's it.
All right, sir. And
Sir,
oxidative phosphorylation, right?
Right.
Is there any need to write the properties of genetic code, sir?
Characters?
Yes, sir. Properties.
No need.
No need. Only need to draw the table.
Draw the table. And also you mentioned 64. And what are the nonsense codons, stop codons?
Yes, sir. Sir,
That's
nonsense codons
Yes,
are
sir.
nothing
We
but
need to
stop codons,
explain
right,
the
sir?
thing.
Both same. Nonsense, stop codons
Stop.
are same. Does not
Opal,
code any.
ochre, amber, right, sir? 3 codons.
Right.
Opal, ochre, amber.
Yes. Does not code for any amino acid.
Yes, sir. They are nonsense.
Yes.
Sir, can we call it as antisense codons?
Antisense codon is part of tRNA.
tRNA structure, right?
tRNA structure, which actually antisense codon carry the amino acid towards the mRNA.
Okay, sir. Complementary amino acid.
Actually, antisense complementary to the mRNA triplet codon.
Right, sir. Right, sir. Okay, sir.
Then, and transcription important for ACA, translation important to ACA.
Okay, sir.
But not completed. We will discuss next class.
Okay,
and all
Okay,
of you,
sir.
please submit your assignment before 20 as early as possible.
Okay, sir.
Okay, thank you all.
Thank you so much.
Okay, sir.
Thank you, sir.
Thank you,
Sir,
sir.
do we need to come there and submit, or can we send it through someone?
You better to give by
Personally?
hand. Yes,
Sir,
personally, better.
సార్ ప్రాక్టికల్స్ ఉన్నప్పుడు తీసుకురావాలని చెప్పారు కదా సార్ అసైన్మెంట్.
లేదు ఇప్పుడు వాళ్ళు ఖచ్చితంగా ముందే ఇవ్వాలని చెప్పారు. ముందే ఇవ్వండి, 20 వరకు ఇచ్చేసేయండి.
Okay. So can we come and
Okay,
submit
sir.
it on Sunday also? Is it possible?
No.
Weekdays
Sunday not
only?
working day. Only weekdays.
Okay, sir. Sure, sir. Thank you, sir.
Okay,
Okay.
sir.
Thank you, sir. Thank
Good
you,
night,
sir.
sir.
Bye-bye.
Thank you, sir.
Thank you, sir.
Bye, sir.